Hello. Welcome to our next lecture on in Cognition. We'll be talking today about cognitive neuroscience. In this first portion, we'll be talking about a simple basic introduction to neuroscience, talk about different levels of analysis in the brain, different aspects of neuroanatomy from the neuron up to the cerebral cortex, and about the ways in which all of this works. Mostly hitting on the stuff that's most important to what we'll be talking about throughout the term. First thing to understand about neuroscience is there are many different levels of analysis at which we can examine the brain, from the molecular, how chemicals interact with one another in the brain, how molecules on the surface of a neuron interact with neurotransmitters, calcium-gated ion channels, etc. Very important part of understanding the building blocks of neuroscience. From there we can move up to cellular neuroscience, looking at how cells function, how they interact with one another. Those, of course, then get built up into neuroanatomy and neurophysiology. We start looking at systems and how they interact. And then we get up to looking at functions. So we look at things like neuropsychology, how individuals with different brain disorders function, and then neuroscience, looking at all of this together and how it works. In particular, cognitive neuroscience, we're, of course, interested in how cognitive functions are related to brain functioning. So we're going to start off with some basic anatomy and physiology, starting off with neurons and neurotransmitters. Neurons are, of course, the individual cells within the brain that create all of our thinking, all of our memory, all of our perception, keep us breathing, feeling, thinking. All of that is neurons and the way they work. Neurons are these individual cells combined together. We call those nerves. So those are bundles of neurons. Put those together and we end up with nervous systems, which are, of course, systems of nerves and neurons. We'll talk about the different subdivisions of the nervous system as we go along. Basic structure of a neuron is relatively simple to get started with. The receiving end of the neuron is what we call the dendrite. These are these bushy projections out from the cell body, which is also called the soma. The cell body contains the cell nucleus and is the basic working center of the cell. Many neurotransmitters are manufactured here and then taken down through the axon down to the axon terminal buttons. Some people call them the terminal boutons, but I think terminal buttons is a little easier, so that's what I call them. Anyway, so the dendrites are the receiving end of the neuron. That's where neurotransmitters bind and change the electrical potential of the membrane of the cell, then generating what's called an action potential. That action potential then travels down the length of the axon, reaching its end, and at the terminal buttons signal the release of neurotransmitters, which cross over the synaptic clefts to then interact with the dendrites of the next neuron. So the sequence of neurotransmissions go from the terminal buttons to the next axon. So the first, or sorry, not the next axon. Neurotransmitters are released by the terminal buttons. They then bind with the dendrites, which are the receiving end of the neuron, that then can generate an action potential, travels down the length of the axon, causing release of neurotransmitters, to then interact with the next neuron. So it's kind of a handoff process. So this is what a neuron looks like in real life. Looks like a pyramidal cell, which gets its name by its pyramid shape. You can see the branching dendrites and then the single long axon coming out from the cell body. So we'll start by talking about the cell body. This is where the cell nucleus is located. This is also where the metabolic center or power source of the cell is, which is, of course, the mitochondria. All of this is there in the cell body. And the cell body is surrounded by a semi-permeable membrane, which allows for ions to travel back and forth. Actually, the entire neuron is covered in semi-permeable membranes, which allow for ions to travel back and forth across that membrane. The dendrites then, of course, are the receiving end of the neuron. They're those short, bushy fibers that branch out from the cell body. These are associated with the postsynaptic neuron. So the synapse is the space between two neurons. So it's between the terminal button of one neuron and the dendrite of another. Information is transmitted by neurotransmitters across that synapse. And so the dendrite is associated then with the postsynaptic neuron. The axon then is a single long fiber that extends from the cell body. It conducts signals from the cell body to other parts of the nervous system. This is associated with the presynaptic neuron. So again, this is on the beginning side of a synapse. Neurotransmitters are released into the synapse, which then bind with the dendrite in the postsynaptic neuron. Axons can be millimeters long to several feet long. So some of the longest axons will travel from the base of your spine all the way to the end of your toe. So it would be a single long neuron that travels the full length of your leg, transmitting information to and from your feet and your toes. The terminal buttons are the endings of the axon. This is where neurotransmitters are released. So neurotransmitters are bundled in vesicles. When an action potential reaches the terminal buttons, it causes the binding of calcium. Those calcium-gated ion channels causes the opening of calcium-gated ion channels, which causes the synaptic vesicles to fuse to the membrane and release their neurotransmitters into the synapse through a process called exocytosis. So those neurotransmitters are dumped into the synapse, then causing their effect on the postsynaptic neuron. Myelin is this white sheath that covers the outside of the axon. This is an important part of functioning of axons because it speeds up the neural impulses. So essentially what happens is, rather than having to travel that electrical impulse, rather than having to travel the full length of the axon, it jumps from what's called the node of Ranvier. So this is a process called saltatory conduction. So this gap in between the myelin sheaths, this is called the node of Ranvier. So rather than traveling the full length of this axon, this action potential jumps from one node to the next, again through this process called saltatory conduction. Myelin is incredibly important for functioning of our nervous system. It's how our sensory and motor systems work. And multiple sclerosis is actually a disease which attacks the myelin sheath. It's actually a demyelinating disease and can cause some pretty devastating symptoms. So as I was saying, give you a closer look at this idea of saltatory conduction. Those signals travel from one node to the other. So you can see here these ion channels open and close in these depolarized regions. So rather than having to travel this full length, it just jumps and then depolarizes the next region and jumps to the next depolarized region. So it speeds up dramatically the process by which this happens. And as I was saying, this is critical for motor functioning. And multiple sclerosis is a demyelinating disease. For some reason, the immune system attacks the myelin sheath and starts breaking it down. So neurons communicate through a process called synaptic transmission. They relay information by the release of neurotransmitters. It's called synaptic transmission because information is relayed across the synapse, which is that narrow space between the terminal button of one neuron and the dendrite of another. So that synaptic transmission is this process by which neurons communicate from one another by releasing neurotransmitters across the synapse. So here's a closer look at how this works. So here we have the axon terminal or terminal button. The action potential reaches the terminal button, causing these vesicles to fuse with the membrane, releasing neurotransmitter into the synaptic cleft. Those then travel across to the dendrite and bind with specific sites, and then have their effect on the dendrite of the receiving neuron. As I said previously, the axon and its terminal buttons are associated with the presynaptic neuron, the dendrite with the postsynaptic neuron. The neurotransmitters released by the presynaptic neuron are chemicals of varying types. We're going to focus on a few of particular importance in this class. We're going to talk a lot about dopamine. It's particularly important for things like working memory. We'll talk a little bit about serotonin. We'll talk about gamma-aminobutyric acid, or GABA. GABA is an important inhibitory neurotransmitter, where GABA is having strong effects. We get reductions in neural functioning, and as a result, GABA can have negative effects on memory. So drugs like benzodiazepine sedatives and alcohol are what we call GABA agonists, so they boost the efficacy of GABA, and as a result, you get reductions in memory performance. We're going to also talk about steroid hormones and their effects on cognition as well. So this whole process occurs primarily through the generation of action potentials. And action potentials are a neural impulse in which an electrical charge travels down the axon of a neuron, which causes the release of neurotransmitters. The action potential is created by the opening and closing of ion channels on the membrane of the neuron. We won't get into the great details of that. Much of that you should have gotten in introductory psychology. But essentially what happens is the cell membrane depolarizes. Once it reaches a certain threshold, it generates an action potential, causing the release of neurotransmitters. Some important points about action potentials. This is an all or none process. That is, an action potential will occur or it won't. Once the synapse has enough neurotransmitter, that is, once the cell membrane has depolarized past its threshold, the neuron will fire. It will keep firing if there is sufficient neurotransmitter present. This level of neurotransmitter is called that threshold. So if there's still enough neurotransmitter beyond the threshold, that neuron will keep firing. That process is then stopped through a variety of processes. One of them is called reuptake. This is where the presynaptic neuron takes the neurotransmitters back into it and recycles them. There are enzymes in the synaptic cleft that break down neurotransmitters, so that's enzyme deactivation. And autoreception occurs. The neurotransmitter binds to the presynaptic neuron and stops the release of additional neurotransmitters. One of the things we'll talk about is how drugs affect cognition, and varying psychoactive drugs have effects on different processes like this. So most antidepressants are selective serotonin reuptake inhibitors, so they block the reuptake of serotonin in the presynaptic neuron. So we'll talk a little bit about that in a variety of topics under the semester. So that's the basic process of neurotransmission. Again, very simple. There's a lot more to this, but that's the level of detail that will really allow you to kind of get through this semester. I do want to talk about the divisions of the nervous system. So we have a nervous system, which is divided into the central and peripheral nervous system. The central nervous system is the brain and spinal cord. Everything else is the peripheral nervous system. Peripheral nervous system includes the somatic nervous system and the autonomic nervous system. The somatic nervous system is the part of the peripheral nervous system that interacts with the environment. So it's called the body nervous system, but essentially it's all of our senses and all of our motor coordination. So all of our sensory motor systems are part of the somatic nervous system. The autonomic nervous system is concerned with regulating the body's internal environment. So heart rate, respiration, blood pressure, skin conductance, pupils, all of that are guided by the autonomic nervous system. The autonomic nervous system includes two different components, the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is our body's arousal system. So when it's activated, we're generally in an aroused state, so a fight or flight kind of response. The parasympathetic nervous system is what's sort of operating under sort of normal circumstances. So we'll take a closer look at that autonomic nervous system. We'll also talk a little bit about the endocrine system. The endocrine system has some important components that will be relevant to our discussions as well. So we talked, as I was just describing, about the peripheral nervous system, in particular the autonomic nervous system. This becomes really important because things like attention are dramatically affected by the activation of a sympathetic nervous system response. And also, certain drugs are what we call sympathomimetic drugs. So things like amphetamines mimic the sympathetic nervous system and essentially trigger a sympathetic nervous system response. So a number of things happen in this kind of fight or flight state. Your pupils will dilate so you can gather as much light as possible. Your bronchi will relax so that you can exchange oxygen very quickly, so essentially it gets more oxygen into your bloodstream. Your heart rate will accelerate and strengthen, so your heart will beat faster and stronger, so it will be pushing blood very quickly out to your muscles. Your internal organs, the blood supply will constrict to the internal organs, your stomach, your intestines, so you won't be hungry, you won't have to pee. You won't be able to get sexually aroused at that time because all of that blood is going towards your muscular system. The whole thing is designed to provide as much energy as possible in that fight or flight situation. Under normal circumstances, your blood vessels will dilate in your internal organs, your stomach will activate, heart rate will slow down, your bronchi will constrict, and your pupils will contract. The endocrine system is often thought of as the body's slow nervous system. It's controlled by the release of hormones. So rather than affecting behavior directly through neurotransmission, we get the effects on behavior, the release of hormones into the bloodstream. So it's a little bit slower, but nonetheless as effective. So hormones come from a variety of places, from the pineal, hypothalamus, and pituitary glands in the brain, the thyroid and parathyroid in the thoracic cavity, the adrenal glands, which sit atop the kidneys, the pancreas in the abdomen, and then of course the sex organs, the ovaries, and the testes, all have effects on various parts of the body and have potentially various effects on behavior. So most of these hormones work in a feedback loop, and I've simplified this a bit, but to give you an idea, there's actually a couple of steps in this. They include follicle-stimulating hormone and luteinizing hormone. But essentially, we have the release of luteinizing hormone, travels down to the testes. Testes then release testosterone. That testosterone then travels back up to the hypothalamus, which stops the hypothalamus from releasing luteinizing hormone. And again, I've simplified this a bit. So then levels of luteinizing hormone drop. Testes stop releasing luteinizing hormone. Those levels drop at the hypothalamus. Starts releasing luteinizing hormone again. Testes release testosterone. That travels up and shuts off luteinizing hormones. So essentially, this is a whole feedback loop that's constantly occurring. So we'll talk a little bit more about sex steroids and how they affect behavior later in the semester. So moving on to the central nervous system, particularly the brain, we'll talk a little bit about organization and function. The first is the cerebral cortex is divided into two hemispheres, the left and right. Each hemisphere is then divided into four lobes: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. So it looks something like this. The frontal lobe is towards the front. The temporal lobes are next to your temples. The very back of the brain is what's called the occipital lobe. That's entirely devoted to vision. And then the parietal lobe, which is located between the occipital lobe and the frontal lobe, up above the temporal lobe. Looking at from the central sulcus side, you can see the frontal lobe kind of wraps around some of these subcortical structures. So the frontal lobe is really a big part of our cerebral cortex. The parietal lobe and occipital lobe are much smaller, and then of course we have here the temporal lobe. So those are the four lobes of the cerebral cortex. We'll get into some of their functions later on in the term. There are some subcortical structures that I do want to talk about briefly. The basal ganglia. These are a series of structures that are involved in motor coordination and implicit memory. We're going to talk a little bit about those again later on in the term, so keep that in mind. The limbic system is something we'll talk quite a bit about. This is a series of structures that are involved in memory, emotion, and cognition. And so we're going to talk in more detail next about the specific structures in the limbic system and how they function. So the limbic system includes the amygdala, which is this almond-shaped structure here. It's involved in some of our sort of primal emotions: aggression, fear, fight or flight emotions. We'll see later on in the term how using a drug called propranolol, you can actually disconnect your fear memories from memories. So people who are, for example, arachnophobic or afraid of spiders, in a single dose of propranolol, they've been able to demonstrate they can actually disconnect that fear. So while the memory of them being afraid of spiders is still there, the fear emotion is no longer present. So we'll take a look at that. The hypothalamus is an important part of the limbic system. It's primarily involved in things like homeostasis, releasing of hormones, and also our feelings of pleasure and reward through the release of dopamine. So when we talk about the reward pathway, it will be important. The hippocampus is what we will probably talk about more than any other part of the brain. This is located below the temporal lobe. We'll talk about the hippocampus and what's called the medial temporal lobe. So this is the part of the brain that's responsible for creating memories or memory consolidation. So if you have damage to the hippocampus, you will end up with what's called anterograde amnesia, or the inability to form new memories. So the hippocampus is a particularly important structure for the formation of memories. And so we'll talk about that later on in the semester. I want to add one final note, is about hemispheric asymmetry and specialization. Remember that the right side of the cortex is the left side, and the left side of the cortex is the right side of your body. For vision, it's the right visual field and left visual field. So we'll talk about split brain patients and other studies where we can project information to only one hemisphere. So if it's in the right visual field, it's going to go to the left hemisphere. So we're going to talk about Navone figures. And so while something that's presented to the right visual field is something that goes to the left hemisphere. So always keep in mind that visual field and hemisphere are opposite. So if I talk about presenting something in the right visual field, that means that it's the left hemisphere. And the opposite is, of course, true. A couple of tasks tend to be lateralized. So language comprehension and production tends to be more left hemisphere, particularly for people who are right-handed, whereas object and face recognition tend to be processed more in the right hemisphere. Well, that's our very quick introduction to some basic neuroscience. In the next lecture, we'll be talking about cognitive neuroscience methods.